We now begin our consideration of the efferent nervous system. Although this is Chapter 11, in the present semester that we're recording, it turns out to be our Lecture 12, in this odd way of trying to assign a recognizable title to things. But anyway, we previously discussed the sensory or afferent division of the nervous system. And one of the reasons I like this Human Physiology by Dr. Silverthorne, using an integrated approach, is the way of considering things collectively and therefore integrated. So we completed our discussion of the afferent division of the nervous system. Now we consider the efferent division, and in this we consider both the autonomic, the visceral efferent, and the somatic efferent, or motor control systems. So both the involuntary and the voluntary considered together. We'll just have a cursory introduction of the somatic motor division, where the CNS control the skeletal muscles through the neuromuscular junctions will be described. And then later, when we talk about the integration of the muscular system and movement, we'll bring it all back together. For now, though, even though we'll do the same for some of the visceral functions like cardiovascular physiology, we'll bring the autonomic division back at that time, but the majority of this presentation will be regarding the autonomic division of the efferent portion of the nervous system. We'll consider autonomic reflexes, and again, for example, when we talk about the urinary system, the reflexive emptying of the urinary bladder, this issue will be revisited. We'll talk about antagonistic controls, the parasympathetic and sympathetic tones at any one point in time, the control of cardiac and smooth muscle and the glands of in homeostasis, so the effector organs or cells for this motor system, and the agonist and antagonist in research and medicine. We begin then with the role of the autonomic division in homeostasis. So the antagonistic branches, of course, are the brakes, if you want to think of it that way, parasympathetic for rest and digest, to restore body function from an activated state or a state of a high level of activity, thinking of exercising or things like that, where at that high level of activation or activity of the organism in general, the sympathetic nervous system, the sympathetic drive would have been high, or in cases of trying to survive, like fight or flight, there would have been that very high energy action or activity that had occurred. So the gas pedal and the brake in that sense. And as we had described before, we never want to think of the situation under ordinary conditions or even slight extreme conditions where we would think that the sympathetic nervous system would be completely turned off and the parasympathetic nervous system completely turned on. But it is always this counterbalancing effect, and somewhere in this region that I'm encircling is probably where you would see the differential activities of the two systems, and rarely one where it would be all on or all off. Although it does occur in some cases, which reminds us that the patterning of the autonomic nervous system, the two antagonistic branches or divisions, throughout the entire body can be different. In other words, the level where you would fall on this balance line at any one point in time for the digestive system innervation by the autonomic nervous system versus the skeletal muscular system would be different, or to the heart versus to the urinary bladder, or to the ejaculatory system in the male reproductive tract. So remember that too much generalization can be a bit confusing and, in that sense, almost harmful. So we do this still just the same, and I try to point out the details as we proceed. So the homeostasis is a dynamic balance between the autonomic branches. So rest and digest is where you have the parasympathetic activity that's dominating. In the fight or flight, you get the sympathetic activity that dominates, and that's a very safe general rule. The hypothalamus, pons, and medulla initiate autonomic, endocrine, and behavioral responses, so a coordination of the homeostatic responses across the board. So in fight or flight, you definitely would have a behavioral component. If you kick back and relax after you've just eaten dinner, there's a behavioral component to that as well, and diverse differing hormonal effects, like as the adrenal medulla dumping epinephrine into the blood during fight or flight, or pancreas dumping hormones into the blood after consumption, ingestion, right? And the autonomic balance would be appropriate for each distribution. So although your heart rate is lower when you are sitting and relaxing, versus being chased by a dinosaur or exercising, the relative differential between the sympathetic and the parasympathetic drive to the heart might be very different, and is indeed very different than the differential drive to the gastrointestinal system. So in other words, if this is parasympathetic drive to your gastrointestinal system after you've finished dinner, you don't want this differential for your heart rate, because your heart rate would be so low you might pass out. So the heart rate might fall over in this area and rest, versus way out here when you're stressed and things like that. So I'm hoping that comes together. So when we think of the autonomic nervous system as part of the involuntary nervous system, even though I may even have represented it as the involuntary nervous system throughout, we know that it is associated with involuntary behavioral centers throughout the nervous system, the spinal cord for autonomic reflexes that we don't have to think about, and even some of the limbic areas and the hypothalamic areas, and certainly brainstem areas that are involved in circuitry that can bring about a complex type of a behavior. So think of a cat when they, as Halloween is approaching here at the time of recording. A typical picture of the Halloween cat is with its back arched, and the hair on its back standing on end, and its tail extended, and its eyes wide open, and things like that. It's possible to recreate those types of defensive, alerted behaviors by stimulating discrete locations, should be pointing down here, in the hypothalamus, or even sometimes in the pontine area, in animals that have had electrodes chronically implanted. We don't do this kind of research anymore. This is from early literature, where we first began to understand how these parts of the brain that we thought were related to other functions could, all on their own, create a very complex behavioral display. And then the endocrine response, and they started looking at the hormonal profiles, was consistent with that. And then, of course, the autonomic nervous system played its role. So the hair standing on its end is piloerection. That's sympathetic nervous system. Anyway, we get aside from this experimental approach and think of what might have been the input to create this involuntary behavioral response. There must be some sensory input, and if there's a sensory input, there's receptors. Well, if we're thinking of homeostasis as a prime example for the hypothalamus, as far as the osmolarity of the blood, so you're going to get thirsty, then we can think of a coordinated endocrine response and the autonomic response related to the blood pressure, and then a behavioral response to drink some water to allow you to dilute that out and get the sodium excreted. So if you become a little bit dehydrated, the hypothalamic receptors detect the change in the osmolarity of the blood, and from the hypothalamus and lower areas that it innervates, and intersociation with the limbic system and the cerebral cortex, you get the endocrine response, the change in the antidiuretic hormones, so that you do want more diuresis. Excuse me, we're talking about dehydration, sorry. Confusing eating a bunch of salt with dehydration. Anyway, with dehydration, you would release antidiuretic hormones, so there'd be less urine output at the time to try to keep as much water as you can to dilute out your electrolytes. And your autonomic response would be to impact the blood pressure. If you dehydrated to the degree that it was decreasing your blood volume, then you'd have to close down the blood vessels to decrease the size of your container to keep your blood pressure in the normal range. And then, of course, the behavioral response is to seek out water. And then, of course, there can be somatic and visceral sensory neurons that can impinge upon these centers to bring about coordinated responses as well. So visual input can trigger involuntary responses in this context. The startle response is a primary one. You don't have to think about initiating a startle response. That's what makes it a startle response, right? It is involuntary at many of these levels of the visceral responses, anyway. So let's consider these autonomic control centers in the brain. So we're familiar with this midline view, a mid-sagittal section of the brain. We recognize the hypothalamus with the pituitary suspended below it, and the midbrain here, and then the pons, and then the medulla, and the cerebellum sitting over the dorsum of the pons and the medulla. And in the hypothalamus, there's a thermoregulatory center for temperature control, an osmoregulatory center for water balance, and this is sensing and with output to motor systems so that you get the behavioral response, and to the vasopressin neurons, so you would regulate the antidiuretic hormone release. And then eating centers. So the hypothalamus is extremely important for triggering food-seeking behaviors, and also ingestive behaviors, taking the food in, as well as termination of the ingestive behavior. In other words, satiety. And so the hypothalamus is a tremendous area of focus for eating disorders, either eating too little or too much. The urinary bladder, you can see, is represented here in the junction of the midbrain and the pons. And there are two respiratory centers. There's a secondary respiratory center in the upper pons, and then a primary respiratory center in the medulla. And then you can see a couple of blood pressure control centers through the pons and the medulla as well. Remember, when you think of blood pressure control, it's heart rate as well as the innervation of the blood vessels for vasoconstriction. So if you constrict all your peripheral blood vessels, you increase blood pressure. And of course, when we get to the cardiovascular system of regulation of blood pressure, and have talked about blood vessels, we'll revisit this type of discussion. So the hypothalamus, basically, we say is water balance, temperature, and hunger in this context of homeostatic regulation of autonomic, endocrine, and behavior responses. Pons is respiration. Medulla is also respiration, cardiac, vomiting, and swallowing. So now they call it cardiac, but over here they're calling it blood pressure. So remember, they definitely go hand in hand. The autonomic pathways, as we should recall, are a two-neuron pathway. There's one neuron in the central nervous system and then a second neuron in the peripheral nervous system that will innervate the target tissue. So the neuron located within the CNS is called the preganglionic neuron because it is on the CNS side or before the autonomic ganglion, and ganglion we know is a collection of neuron cell bodies and their dendrites in the periphery. And there's an interneuronal junction between the two. The name for the neurons within the autonomic ganglia are the postganglionic neurons. Just to complete the parallel terminology, the only part of the postganglionic neuron that is after the ganglion, of course, is the axon of that neuron, but we don't let that confuse us. And we'll talk about the differing transmitters in pre versus post and for the two divisions of the autonomic nervous system. And we'll discuss the receptors for each as well. So we've talked about gas pedal and brake. So the antagonistic control of the autonomic division. Most of the internal organs are under this antagonistic control. So the visceral distributions of these general visceral efferents, sympathetic and parasympathetic, will really work against each other. But there are instances where you only have one innervation, and typically it's the sympathetic. And that is for example, the distribution of the postganglionic sympathetic axons with the arterial system out into our peripheral vasculature, into the skeletal muscle and the dermis of our skin and the hypodermis. And so you really don't have postganglionic parasympathetic fibers out there. It's just the gas pedal that's going to regulate it. So if you turn off your gas, take your foot off the gas pedal, you slow the car down just as much as if you'd put your foot on the brake, if you want to think of it in that way. Sorry about that. Most internal organs are under the antagonistic control, as we said. One branch is excitatory and the other is inhibitory. Excuse me a moment here. Okay. The autonomic branch is split in that sense, gas pedal and brake. And with the effector organ, the heart, we know very well that the sympathetic drive will cause tachycardia, increase in heart rate, and the parasympathetic drive will cause bradycardia, a low heart rate. The parasympathetic response will slow the rate, and the sympathetic will increase the rate or the force of contraction. So the autonomic, sympathetic, and parasympathetic pathways can be divided anatomically as well into the parasympathetic, where we find the preganglionic neurons in nuclei of cranial nerves, 3, 7, 9, and 10, right? So cranio, and then sacral, S2, 3, 4. So craniosacral. So another name for the parasympathetic nervous system is the craniosacral system. And then the preganglionic neurons for the sympathetic nervous system are in the thoracic spinal cord levels and the first two lumbar, so T1 to L2, or thoracolumbar. So we have the craniosacral for parasympathetic and the thoracolumbar for the sympathetic. And we're going to proceed through a few illustrations to differentiate these two systems, kind of descriptive, not detailed anatomy, but descriptive anatomy, and then we'll get into the function of the different transmitters and receptors. And as you can see from this table, we're going to get into the type of receptors for the sympathetic and nervous system that is shown here, at least. And so we also recognize that there have to be ganglia for the sympathetic nervous system, and it turns out to be the sympathetic chain that runs alongside the vertebra, called the paravertebral ganglia. And then there are some that actually sit in front of the vertebra, or even in front of the aorta, called the preaortic ganglia. And these peripheral sympathetic ganglia will contain the postganglionic neurons whose fibers will jump onto the arteries to be distributed to the viscera. So the detailed anatomy will not be part of this discussion, and I refer you to an anatomy text for that. The parasympathetic ganglia, we already considered these in the head region and discussed them in a previous presentation, and then we note that in all the other target sites, they're showing the postganglionic neurons distributed right within them. Let's see if the kidney is not. So the first obvious difference between the two of these is that the parasympathetic postganglionic neurons are very close to the effector cells, very close when in comparison with the sympathetic postganglionic neurons. So let's take a look at this table then, and we'll proceed through some more detailed descriptions. But obviously, from this list, you see that there are sites where you get both parasympathetic and sympathetic innervation, and then the kidney and the adrenal medulla here were a couple where we said that there was just the sympathetic innervation, but not the parasympathetic. And what they're not showing here is the sympathetic postganglionic fibers going back into the typical spinal nerve and being distributed with the spinal nerve and then getting onto arteries either there or somewhere close to here, such as up here they'll get onto the common carotid artery and be distributed with the arteries to all the target cells and the vasculature in the head and neck region. So the vasculature is an example of sympathetic innervation without a corresponding parasympathetic innervation. So the effector organ lists the pupil of the eye through lymphoid tissues, which are not illustrated here. And we'll go through each of these and then talk about the parasympathetic response, the sympathetic response, and the adrenergic receptor that would be found there. So the pupil of the eye was considered in the previous lecture series on sensory physiology, where the pupillary reflex was mentioned. So with the parasympathetic activation through the oculomotor nerve, remember 3, 7, 9, and 10 carry the parasympathetic preganglionic fibers. So this would be cranial nerve 3 here, and the parasympathetic innervation of the pupil of the eye would cause constriction of the pupil. The sympathetic response would cause dilation. So if you relax the muscles, excuse me, then the pupil will. Oh, this is. Sorry, this is going to cause a constriction, and then the sympathetic is going to cause the dilation. So it's the opposite of what you think, and I got caught in that myself, right? That was because I was reading below here for the double asterisk. All parasympathetic responses are mediated by muscarinic receptors, as we had talked about before. So muscarinic receptors will cause the constriction of the pupil of the eye. And the alpha adrenergic receptor will cause dilation of the pupil of the eye. For the salivary glands, there's a watery secretion, of course, and that is stimulated by the parasympathetic. The sympathetic will cause the secretion of mucus and certain enzymes through the alpha and the beta-2 adrenergic receptor. The heart is slowed, bradycardia with parasympathetic, and tachycardia, and increased contractility or force of contraction by the sympathetic mediated through the beta-1 adrenergic receptor. Arterioles and venules here on the table shows us that there is no corresponding parasympathetic response because there's no parasympathetic innervation. And depending upon the type of receptor that would be present on the target cells, the smooth muscles, you either get a constriction through the alpha adrenergic receptor or a dilation through the beta-2 adrenergic receptor. So you have to know where the receptors are distributed on the vessels, not so much whether or not they receive the sympathetic innervation, because all vessels receive sympathetic innervation, so it's the smooth muscle cells there that express different receptors to determine the response to sympathetic activation. And as well, in the lungs, the parasympathetic response is a bronchiolar constriction. You don't need the large volumes of air moving in and out of the lungs when you are resting and relaxing, but when you are being chased by a dinosaur, you want bronchial dilation brought on by the sympathetic innervation, and that's through the beta-2 adrenergic receptor. And we note below here there's the hormonal epinephrine only that is bringing this about. That's the single asterisk footnote. In the digestive tract, increased motility and secretion is the parasympathetic, decreases motility and secretion through the alpha and the beta-2 receptors for the sympathetic. For the exocrine pancreas, you increase enzyme secretion, rest and digest, with parasympathetic, and that is turned off. That's a break through the alpha adrenergic receptor. So remember, you can achieve a decrease in enzyme secretion by turning off the parasympathetic drive. Same thing with the heart rate, is that you can decrease parasympathetic activity to the heart, and then the relative sympathetic tone, even though it didn't change, but relatively speaking to the prior level of parasympathetic activity, it is now higher, it's greater than it was before, and it takes over, and you still get an increase in heart rate through the beta-1 receptor, but not because of a higher sympathetic activity or tone, it's because you took your foot off the brake. So this can be, wherever you have both divisions represented, this can occur. In the endocrine pancreas, the parasympathetic, you want insulin secretion. With the sympathetic, it's going to decrease that through the alpha adrenergic receptor. The sympathetic response of the adrenal medulla is to secrete catecholamines, epinephrine being the primary one, but norepinephrine and dopamine will also be part of the secretion, but not the dominant form of the catecholamine. In the kidney, we saw it was just the sympathetic nervous system. We're going to get, through a beta-1 adrenergic receptor, the increases in the renin secretion. In the endocrinology chapters, we talked about the renin-angiotensin system, and when we get into the urinary system and blood pressure regulation as an integrative approach, we'll bring the renin-angiotensin system back into our discussion at the time. In the urinary bladder, but another very important correlative of this impact on the kidney comes back up to this general one here with the arterioles. The alpha adrenergic receptor is expressed by the smooth muscles of the renal arteries and arterioles, and activation of the sympathetic nervous system is going to cause constriction of the renal vessels, renal arteries and arterioles, and at the same time you're going to get an increase in the renin secretion. So two ways that the function at the kidney can be, to use it as a prime example, can be affected by the sympathetic nervous system through its vasculature. and through this endocrine system. The urinary bladder, release of urine, you have to get an increased activity of the cervical spinal cord to, they're not showing it going to the urinary, well, they're showing it going to the urinary bladder, but you also have to relax the urethra, the urethral sphincter, right? So that's part of what's being considered here. And then with the alpha and beta 2 adrenergic receptors, you get urinary retention. So when you're running away from a dinosaur, you don't want to leave a trail of pee. So you're going to constrict your smooth muscle. It's called the internal urethral sphincter. We know we have a conscious one, an external one too. Same thing with the anal sphincter. That is skeletal muscle, and that's the one that we control when we're holding it in, but the reflex, the autonomic reflex, is saying we're ready to pee, and consciously with your external urethral sphincter, typical neuromuscular junction, you are keeping that skeletal muscle sphincter constricted, right? So again, remember, this would for the urinary bladder, what we'd be talking about here is a reflex function of turning off sympathetic and turning on parasympathetic, is an involuntary reflex at the level of the sacral spinal cord that we don't control. It's the external sphincter that we consciously control. So think of a child growing up. They have to be trained to control the voluntary or external urethral sphincter so they don't pee in their pants and stuff like that. But the visceral one is involuntary, and it continues throughout life. So all we can control is the somatic urinary sphincter. Same thing with the anal sphincters. So adipose tissue selectively receives a sympathetic innervation through the beta receptor, and the textbook mentions that this is a beta 3 receptor, a unique type that's just on the adipose tissue for fat breakdown. And the sweat glands, for localized sweating, the sympathetic innervation, so the vasculature, or the typical peripheral spinal nerve, that had received the postganglionic from the sympathetic chain will go on out. Picture it going on out to your fingertip, and you get the innervation of the sweat gland. The male and female sex organs, so the parasympathetic is erection, vasculature, and the sympathetic is ejaculation in the male, the contraction of smooth muscle in the ejaculatory ducts, or the ejaculation-related ducts. There's the vas deferens and a series of them if you are familiar with the anatomy, and that's through the alpha adrenergic receptor. The uterus depends on the stage of the cycle, the effect of the parasympathetic and the sympathetic. The sympathetic is mediated through the alpha and the beta 2. With the lymphoid tissues, generally the sympathetic nervous system is inhibitory through the alpha and the beta 2. Thank you for sticking with me to get through that table. Well, now let's think about the pathways. First we'll get through the text, and then we'll consider this. This is the vagus nerve, just one of the cranial nerves carrying parasympathetics. Remember 3, 7, 9, and 10. The vagus is the 10th. So this is that last cranial nerve that would be carrying the parasympathetics to the viscera. After that, the distribution would be through the sacral, the pelvic, splanchnic nerves. So the sympathetic versus the parasympathetic pathways, the spinal cord exit, the neurotransmitters and the receptors will be considered. The major parasympathetic tract is the vagus nerve, therefore vagrant. It's a wandering nerve. So it's not all that often described with these illustrations, so I'm going to take you through some of it here because it is responsible for the autonomic control of the mucus secretions throughout the pharynx, but also skeletal muscle control for the soft palate, the pharynx, and then ultimately to the esophagus for the swallowing reflex. So the vagus nerve is critical for respiration and ingestion, right? It is also responsible for the triggering. You see that it innervates the stomach eventually, for triggering the reverse peristalsis up through the esophagus for vomiting. So remember we said that the vomiting center was in the medulla, so local innervation of the vagal motor. There's still visceral efferents because they're going to visceral structures, even though it's skeletal muscle, and can regulate very quickly the motor portion of the vomiting reflex. But even though they're showing it going to the musculature of the soft palate, the pharynx and the larynx and the esophagus, remember that the parasympathetics will be delivered with it, and the postganglionic neuron is going to be very close to the target site, the mucosa, where it's going to innervate the mucosal glands. And as well, you see here the sensory innervation of the carotid sinus and the carotid, no, not the carotid body, that's cranial nerve 9, sorry, the carotid sinus here to pick up pressure. So it's already also the general visceral afferent that's within here, and that's why we see the two sensory ganglia hanging off the vagus close by. So there's more being represented here than what is implicated in the context of the title for this slide. And one other thing I want to point out, this would be on the left vagus. You can see a recurrent laryngeal nerve here that comes up and innervates the internal muscles for the voice box. So it's through the vagus nerve that we can control the voice box for speech. Of course, other nerves are involved in the oral cavity portion of it, but for the voice box itself, the vagus innervates it. But as well, any of the mucus glands or parasympathetic innervations along this are going along with the vagus there. In addition, we can see how branches of the vagus can come off and innervate the arch of the aorta, and of course come in here to innervate the SA node in the right atrium. The reason for talking about the innervation of the right atrium and the arch of the aorta is not that we have parasympathetic innervation there. It's not what it is. It's general visceral afferent stretch receptors. So when the atria stretches, you know that there's more blood coming back to the heart. When the aorta stretches, you know that there's a greater volume of blood being pushed out of the left ventricle, and that sensory information is brought up for the integration of the cardiac center in the medulla. Right? So you have to have sensory input to have a visceral reflex. So part of the visceral efferent response to this type of change in pressure would be to affect the preganglionic parasympathetic output of the vagus nerve. So if there was an increased blood coming back into the right atrium from the systemic circulation, then the heart is going to receive more blood, and you will, through this reflex, decrease the parasympathetic drive and maybe increase the sympathetic drive a little bit. And what this will do is temporarily increase heart rate to get that blood through there faster and more effectively. We also see the sensory and parasympathetic innervation to the lungs, although there's more sensory than parasympathetic. And then once we get back to the vagus, where it continues on the esophagus into the abdomen, onto the stomach, and then to the liver and the pancreas and the small intestine and the first part of the large intestine, other than general visceral afferent sensory, right? Because you can tell when you got a big pocket of gas in your colon, right? And you can feel it gurgle across here. That's sensory information coming all the way up there to the vagus. Or when the stomach is stretched, that's supposed to be the vagus information to be signaled to the hypothalamus that you're full and you should stop eating. So the parasympathetic, what we're thinking about is the parasympathetic innervation of the glands of the stomach. So somewhere nearby here have to be postganglionic neurons of the parasympathetic system. And remember, we talked about the enteric nervous system. The enteric nervous system is where we find the parasympathetic postganglionic neurons, right in the wall of the gut tube. Excuse me. So I won't go on anymore now. I hope that helps you understand the vagus nerve and puts it into an integrative perspective at this time, where we talked about integrated endocrine, innervation of the pancreas, but you have to have a sensory input, right? So when the stomach stretches, then the vagus knows already, if you want to conceptualize it that way, that it reflexively should be triggering release of hormones from the pancreas, and as well the digestive enzymes. There's other local stimuli that end up being endocrine to contribute to that, as you know, and direct effects of the absorbed glucose on insulin. But this is the vagal portion of it, early in the ingestive and digestive stages. So this is a great time just to take a moment and think about the vagus nerve and everything that it does, and then we'll revisit it later. So to go back just to the autonomic nervous system, it is considered to be only an efferent nervous system, even though there's definitely a sensory component, as we just described. Typically the sensory component, the general visceral afferent, is not included in the autonomic nervous system. It's just one of those weird things. You need to know it, but it's not such a big deal. We still understand the autonomic motor system as a two-neuron system. There's the CNS and the PNS component. So in each case we have that, and I think we're going to go through... I thought there was a set of slides where they split these out. So we'll go through it on this slide. So there's the preganglionic sympathetic neuron that synthesizes and secretes or releases acetylcholine onto the postsynaptic neuron, which would be the postganglionic sympathetic neuron in the sympathetic peripheral ganglia. And the cholinergic receptor on the postganglionic neuron is the nicotinic receptor. Well, this is the exact same for the parasympathetic preganglionic onto the postganglionic parasympathetic neurons. The parasympathetic postganglionic neurons are in completely different ganglia than peripheral ganglia than are the sympathetics. As well, when you get into the intestine, when the postganglionics are in the wall within the enteric the enteric nervous system, you do not find sympathetic postganglionic neurons there. Their axons will be there, going to the gut, just like the parasympathetic postganglionic ones. So still, on this point, that the parasympathetic postganglionic neuron cell bodies are never, I'll feel confident in saying that, found at the same location as a sympathetic postganglionic neuron. They're always in different locations. But their terminals, when they converge on common targets, will be very close to each other. And what will differ, however, as you know, is that for the sympathetic nervous system, the postganglionic neurotransmitter is norepinephrine. And then we've talked about the different types of adrenergic receptors. And for the parasympathetic postganglionic neurons, they will synthesize and release the same transmitter as both preganglionics. They are also cholinergic. And what differs is that the postsynaptic receptor is muscarinic type. And the nicotinic, as you hopefully recall, are ligand-dependent ion channels or ligand-gated ion channels, sodium, and therefore they depolarizes the postganglionic neurons. Same here. But the muscarinic is a metabotropic type of a receptor, as are all of the adrenergic receptors. So thus, you recall that you can have the metabotropic receptors affecting ion channels, so you can get an immediate response. May not be as fast as these fast ligand-dependent ion channels, but you can get a recognizably immediate response. But then there's going to be a secondary phase, because it's activating enzymes and stuff like that, second messenger systems, that will be more protracted. So this is the benefit of the autonomic nervous system. You need to have an immediate increase in heart rate when you see the dinosaur, right? And you have to sustain some of the changes over the period of time it takes you to escape and recover from being chased by a dinosaur. So the autonomic nervous system classically has that early change that we think of as immediate, but it's not as quick as a neuromuscular junction to twitch a skeletal muscle. But then you get a prolonged effect, whereas your skeletal muscle is going to twitch, contract, and then it would stop. It wouldn't be a lingering effect. So in the evolutionary sense, we can see how there would be an adaptive advantage to having the metabotropic type of receptors in the target tissues for the autonomic nervous system. Well, let's think a little bit more about the postganglionic autonomic neurotransmitters for the sympathetic division and the parasympathetic division. As we said, they differ. It's norepi for the sympathetic and acetylcholine, or ACH, for the parasympathetic. Alpha and beta adrenergic receptors versus nicotinic and muscarinic. The catecholamines, which includes norepinephrine, are synthesized from tyrosine, and as you recall, acetyl-CoA and choline are the precursors for the synthesis of acetylcholine. Remember, it was choline acetyltransferase was the enzyme to synthesize this, and then there was a whole series of enzymes to convert tyrosine ultimately to dopamine, then to norepinephrine. And then in the adrenal medulla, there's one final enzyme that will convert the norepinephrine into epinephrine. But each of these also has to be inactivated, so the acetylcholine esterase we'll see repeatedly right in the synaptic cleft, breaks it down into choline and acetate right there in the synapse. And then the monoamine oxidase will be in the mitochondria of the axon terminal, or the varicosity, that released the norepinephrine. So we have to have norepinephrine reuptake transport protein present as well at these sites. What they're not mentioning here is that there is also an extracellular degradative enzyme that will break down the catecholamines once they've been released. So as we said, there would be the transport proteins for norepi here, and then for choline at the parasympathetic sites. So with the autonomic targets then, we think of the pathways controlling smooth muscle, cardiac muscle, exocrine glands, select ones, and endocrine glands, only select ones, not all of them, lymphoid tissue, and adipose tissue. The autonomic neuron structure, we're thinking of the neuroeffector junction. The postganglionic axon won't form the same type of axon terminal that we considered with the central nervous system, or even that we'll see at the neuromuscular junction. These are little dilations, so it looks like a beaded necklace where the bead is a varicose portion of the axon called a varicosity. And the axon is where we will see the neurotransmitter synthesis. So the question I ask you at this point is that this is just a classical axon. We can see it having the varicosities along its length, and the synaptic vesicles containing the neurotransmitters distributed within there, as you'd find pretty much in any axon. And we need to ask ourselves, how could we convert this varicosity, a little swelling where we've got some of these vesicles accumulated, into a site of release? We know action potentials are going to be conducted down the axon. How do you convert this into a site of release? Well, you need the voltage-gated calcium ion channels there, right? So typically what is happening is the target cells, the effector cells, are communicating with the axons in areas to trigger changes such that the voltage-gated calcium ion channels will be deposited here. Remember, they have to be shipped in and out by the neuron cell body. So all the way back and forth, you've got anterograde and retrograde transport, just as we described for any other axon. And all this membrane has to be supported. The phospholipid bilayer and the protein contents maintained by the soma. So something tells these varicosities to accumulate voltage-gated calcium ion channels here, such that when you get action potentials coming down through, you get vesicular release of norepinephrine. And typically that's because the target cell is there and it's signaling it, saying, I'm here, this is where you need to set up this special type of structure. So we see that with the smooth muscle cells being distributed in the wall of the gut tube, in the wall of an artery or vein. Of course, they'd be modified cardiac myocytes in the sinoatrial node, or they would be cardiac myocytes in the muscular wall of the heart, things like that, anatomy that we'll consider later. But regardless of what exactly, or glandular cells, if it's a neuroglandular type of junction, anyway, you're going to see these varicosities. And the site of release is along one of these varicosities, and they have to have the voltage-gated calcium channel there, because when the action potential reaches it, that will open, and it's that calcium-dependent exocytosis that is responsible for the localized release of the neurotransmitter, in this case norepi. So the tyrosine is acted on enzymatically through a series, ultimately until norepinephrine is produced, and that norepinephrine is accumulated in the vesicles. Upon release, it diffuses, not within a synapse, but just in the extracellular interstitial fluid here. It's available, it's bouncing around, so it's going to bounce into the presynaptic membrane. If it bumps into the norepinephrine transporter, then it'll be taken back up, and here it can either be used to recharge the vesicles for re-release, or the monoamine oxidase that's here at the mitochondria will act on it. And it all depends on, it's concentration dependent in here. It's bouncing around, bumps into the transporter, it'll get transported into the vesicle. If it happens to be one that bumps over here into the MAO, then it's going to be degraded. So the action potential arrives at the varicosity. The depolarization opens the voltage-gated calcium channels. You get the exocytosis that is triggered. Then the norepinephrine diffuses, and some will bump into the target cell membrane where the receptor, the adrenergic receptor, will be located. They're G-coupled, coupling it to the response in the target cell. The receptor activation ceases when the norepi diffuses away from the synapse. So it's diffusing through here throughout this entire time. So it's an ongoing process. If you think of this interstitial space, similar to the synaptic cleft, as a bathtub where you got the drain open part way here, represented by these two, and the water turned on at a certain rate, then you have a certain amount of water in the bathtub, but it's constantly being changed. Some going in, some coming out. You increase you open the water faucet more, and then you got more water in the bathtub. So try to think of it always as a very dynamic process, not these fixed stages as they tend to do in these textbooks. So the norepi is removed from the synapse in both of these ways, and it's in the process of diffusing about here from high concentration to low concentration, more remote. It's either going to spill over into the plasma, excuse me, and in times when there's a high level of sympathetic activity, you actually can measure the spillover of norepi in the plasma. So the plasma norepinephrine has two sources, the adrenal medulla and the spillover from the collective sympathetic fibers distributed throughout the body. So with a massive sympathetic discharge, you'd get a robust increase here, and any degree of a lesser sympathetic discharge throughout the organism would be represented and detectable by the plasma levels of norepinephrine. But it's one way of removing it from the target site, too. You just wash it away. And then it, of course, can be taken back, as we had mentioned. So if we think of the stimulation of the sympathetic branch, it's going to be the pupillary dilation. So if you're running away from a dinosaur, you want to be able to see as much as you can, so you get pupillary dilation. If you're resting and relaxing, you're going to have pupillary constriction to focus close up on a book in front of you. You're going to sit and read as you're digesting. The sympathetic branch stimulation would induce salivation. That's by increasing the blood flow. But that's part of that differential regulation that we're talking about. You typically don't associate the sympathetic nervous system with the ingestion and digestion, and salivation is certainly linked to that, but it's related to the blood flow. Saliva comes from plasma originally, right? Anyway, put it together to make sense of it there. Heartbeat and volume, blood vessel and bronchiolar dilation, fat breakdown, and ejaculation. Actually, this is an inhibition of salivation, right? So you get vasoconstriction, reduced salivation. You get dry mouth when you've got stimulation of the sympathetic nervous system. And it will inhibit digestion, pancreas secretion, and urination. So I got to go back to the text. I did read the chapter, and I don't recall what it said specifically about salivation, but I'm going to go and check on that. Usually when you're stressed out, you get dry mouth, So let's try to make sense of that. But it definitely will inhibit digestion, shut down the secretion of the stomach and the pancreas secretion, and certainly, as we said, will decrease the urination. Well, with the adrenal medulla, the primary neurohormone, as we said, is epinephrine, and it has multiple and distant targets. So let's consider this. The next series of slides take us through this set of panels, A, B, and C. So we know that it's the adrenal or suprarenal gland sitting on top of the kidney or the rena. And the adrenal gland has the adrenal cortex, where if we start from the connective tissue capsule, as all organs have, you would find most superficially the adrenal cortex with the different layers. And do you remember those? And then the deepest portion is the collection of cells that are produced from the neural crest cells. I believe we discussed the development of the nervous system and talked about neural crest cells. And the adrenal medulla is where we find the modified sympathetic ganglion, as I said, neural crest cell origin. So they're kind of like neurons, and they express the genes for all the enzymes to produce the catecholamines right to epinephrine. So here's the circuitry. This should be up a little bit more in the intermediate horn of the gray matter of the spinal cord for the preganglionic sympathetic neuron, somewhere in the thoracic or lumbar region. It's actually in the thoracic region. And the preganglionic sympathetic neurons, its axon, will be distributed directly to the adrenal medulla, and it will synapse on the adrenal medullary cell. They're calling it a modified postganglionic sympathetic neuron. It's, I guess that's one way of considering it. And through the nicotinic cholinergic receptor, of course, you get the exocytotic event for the release of epinephrine, but it diffuses into the vasculature there and is distributed, as with any hormone, through the peripheral vasculature to its target tissues. The parasympathetic branch is acetylcholine with the muscarinic receptors. As we said, they're G-coupled. The second messenger pathways will bring about the protracted responses, and there's at least 5 subtypes of the muscarinic receptors, so it's a little more complex. The actions would be to constrict the pupils and the bronchioles, slow the heart, stimulate digestion, insulin release, urination, and erections. The autonomic agonists and antagonists are important tools in research and in medicine. So the cholinergic system, we said that there were at least 5 types of muscarinic receptors. I missed the cholinergic up here. We need to remember that there's acetylcholinesterase inhibitors, such as neostigmine, and an acetylcholinesterase inhibitor would cause acetylcholine levels would decrease the rate of removal of acetylcholine from the synapse and sustain higher levels in the synapse. That's why they're useful drugs. I don't think neostigmine is used, but the ACHE inhibitors used in Alzheimer's disease are for that purpose, to increase acetylcholine levels in the synapse in the cortex that are at very low levels to begin with. So whatever the acetylcholine you get released, you want to keep it around as long as you can. As far as warfare is concerned, this is not a good thing, because it would paralyze you, and of course it is of great concern to military personnel, and they're trained how to deal with it. They're given drugs to inject, one of which is atropine, that would be an antagonist at the peripheral target sites, and would prevent this neostigmine induced increased acetylcholine from having its major effect. And then I'm not sure what other substances in there that would block the nicotinic cholinergic receptor, so they wouldn't become paralyzed. Basically what these nerve toxins will do is prevent you from breathing as part of the paralysis, and you suffocate. So not only immobilizes, but it is lethal. And of course the excuse me. It's the bungarotoxin that is used to be injected in the skin cosmetically, right? Am I right there? I'm trying to remember that. Hopefully you did. The triethanolamine ammonium, triethyl ammonium, is effective, and it is selective for the ganglia. And then curare is a classic peripheral nicotinic cholinergic antagonist. And quite obviously, nicotine is the agonist, and muscarine is the agonist, and therefore the names of the corresponding receptor subtypes. For the adrenergic, of course, the neurotransmitter, the endogenous agonists are norepi and epi, and there are indirect agonists or antagonists. The indirect agonists would stimulate norepi release, such as ephedrine or amphetamines, and preventing the norepi uptake would be cocaine. So for the alpha receptors, phenylephrine is a short-acting agonist. If you have ever seen the 12-hour nasal decongestant spray, I'm not recalling the name of the molecule in there, but anyway, it's a more potent agonist, similar to the phenylephrine. So the 4-hour nasal decongestant is phenylephrine. The 12-hour is this other substance, the name which escapes me at the moment. And then there are a wide collection of alpha blockers. Then the beta-adrenergic isoproterenol is kind of a classic agonist, and there's several types of beta blockers, such as propanolol, which is a beta 1 and a beta 2 selective, and then metoprolol, which is a beta 1. So these are typically, the beta blockers, as you might know, are typically given to strengthen the heart rate, the heartbeat, in cases of cardiac failure. So whatever muscle is left, you try to get it to operate as optimally as possible. So we see the efferent pathways into the peripheral nervous system illustrated on this slide. And we'll look at the autonomic pathway here, and then each one separately through there, and then do the somatic. So the make a couple of points first. For the sympathetic, parasympathetic and sympathetic pathways, we are first thinking about the lengths of the fibers for the preganglionic and postganglionic neurons, and therefore relative location of the ganglia. For the parasympathetic system, we said the ganglia was close to the target cells, so the postganglionic fiber is short and the preganglionic fiber is long. So think of the vagus that we talked about, how long the preganglionic axon is all the way down to the colon from the brainstem, so the full length of the trunk of our body and the neck, right? And then the postganglionic is going to be very short, right inside the wall of the colon. Whereas for the sympathetic nervous system, it'll come from the thoracolumbar spinal cord just to the paravertebral ganglion, and then the postganglionic fiber will either get into a spinal nerve and then go out to the tip of a finger, we used as an example. So very long postganglionic, but preganglionic is short. And then, of course, the adrenal medulla we've talked about already, so we won't deal with that until we get to the slide that shows the targets. So the parasympathetic, this is a summary, as you know, with the autonomic effectors, a smooth and cardiac muscle, some endocrine and exocrine glands, and some adipose tissue. So no need to go through all of that. We see, again, the shared acetylcholine for the sympathetic with the nicotinic, and of course muscarinic post for the parasympathetic, but sympathetic, it differs. In this case, we're showing the alpha receptor with autonomic effectors of the smooth and the cardiac muscle, some endocrine and exocrine glands, and some adipose tissue. See that? Okay. And then for the other adipose tissue, they're still not showing the beta-3 receptor as mentioned in the textbook for the adipose tissue. But anyway, the blood-borne epinephrine targeting beta-1 receptors and beta-2 receptors predominantly. So finally, a comparison of the somatic and the autonomic nervous systems. The number of neurons in the efferent path, 1 for somatic, 2 for autonomic. The neurotransmitter is acetylcholine, but the nicotinic postsynaptic receptor. And of course, both nicotinic and muscarinic, as well as the adrenergics for the autonomic. Target tissue, skeletal muscle, versus the ones that we had listed previously. The neurotransmitter is released from a typical type of axon terminal for the somatic, where it was at the varicosities along the target tissues. In some cases, also the axon terminals, such as between preganglionic and postganglionic neurons. It's a prime example. The effects on the target tissue, it's going to be excitatory only, where you get muscular contraction for the somatic, and you can get both excitatory and inhibitory for the autonomic. The peripheral components found outside the CNS, it's only going to be the axons for the somatic, but for the autonomic, you're only going to find preganglionic axons in the periphery, and then ganglia, and then postganglionic neurons, which includes their dendrites, their soma, or cell body, and their axons. In other words, the postganglionic axons can be found in places where the rest of the neuron cannot, right? And then the summary of the function, you get posture and movement for somatic, as we well know, and then visceral function, including movement of internal organs and secretion and control of metabolism for the autonomic. So let's wind up then with this consideration of the somatic motor system, with the CNS origin being in the ventral gray matter of the spinal cord, and then certain cranial motor nuclei, and then we know that they're going to be myelinated, whereas something that we didn't mention with the autonomic nervous system is that the preganglionics are myelinated and the postganglionics are unmyelinated. The somatic motor division, remember, motor is only myelinated axons. The somatic sensory has both myelinated and unmyelinated, but those are peripheral processes of pseudounipolar neurons, whereas here we're just talking about the peripheral axon of a somatic motor neuron, and they're going to be myelinated. The terminus will branch, so it has that telodendria type of a branching, and it'll form a neuromuscular junction. So it would show this long axon from the CNS to the target tissue, the target cell, with the acetylcholine being released and the nicotinic cholinergic receptor being expressed by the postsynaptic or the target cell. And it will be inserted selectively into the membrane, just subjacent to where this widely branching axon terminal, the telodendria, would be occurring, and that's called a neuromuscular junction. Note that the myelin is right up to the pre-terminal portion, if you will, and from there on out you're still going to have the satellite cells surrounding it and other connective tissue cells nearby. Excuse me. So in this case, they're showing the Schwann cell sheath coming up very close to the neuromuscular junction. And the term that we use for the postsynaptic membrane, the modified membrane of the skeletal muscle cell, is motor end plate. And we'll obviously look at this again when we talk about the contraction of a muscle originating from the neuromuscular junction, but we can introduce it at this time. So if we zoom in on that motor end plate and its association with the axon terminal, Then we see the presynaptic membrane with the vesicles that are containing acetylcholine, very similar to what we've been seeing all along. A synaptic cleft, the volume of which is increased by these tremendous infolds of the postsynaptic membrane. These accomplish one other thing as well, and that is they increase the surface area within which you can have these nicotinic cholinergic receptors inserted, and so a very high local concentration of the nicotinic cholinergic receptors. So in other words, the likelihood that any one molecule of acetylcholine getting into the synapse having an opportunity to bump into randomly a nicotinic cholinergic receptor is very high. So you're going to get a very quick and robust response in the target cell. As well, don't forget, distributed along here is going to be the acetylcholinesterase. They're just showing it at the periphery, but I think it's distributed a little more uniformly throughout. So you're going to terminate the signal just as quickly as you can respond to it. So that's why you want all these receptors there. So any acetylcholine coming into the synapse has that opportunity to find one of them and to bind to produce a response very quickly. And then you've got the enzyme right there to eliminate it as soon as it comes off, so you can reactivate that muscle again within a very narrow window of time. So this is how this system has adapted through the evolution of biological systems. So quite obviously, as in all cases, the action potential comes, depolarizes the membrane, voltage-gated calcium channels, calcium-dependent exocytosis, diffusion, doing all the things, bouncing around, and then gets broken down, and then the choline reuptake protein isn't shown here, and then resynthesis and recharging of the vesicles. So one way of thinking about this is you'd need like 2 times as many of the nicotinic receptors at the postsynaptic membrane versus, say, a glutamate receptor, because 2 acetylcholine molecules need to bind to every nicotinic receptor to cause it to open, whereas glutamate just requires 1. Kind of an odd way to just make the point that you really need a high density, and you also require another thing, that that one acetylcholine molecule in the synapse can't just activate one nicotinic receptor. You need a couple of them. So it's still very concentration dependent, but the system is quite well refined to manage this, as we saw, the infolding of the membrane and the high opportunity for for the acetylcholine to have its effect is there. So once it's ligated, then the ligand-dependent channel opens and sodium goes in. Now, these are typically going to allow some potassium out, but more sodium will come in, and so you get depolarization, as you see here with the positive charge inside, the negative charge outside. So this is the final slide, and it is a set of figures from the textbook, and of course it's related to nicotine being legal and readily available to humans for consumption, and having a very potent addictive quality. In the textbook, the current edition at least, I see it as the 5th edition. There are some questions that go along with this, and what I'll do is I'll just read those questions to you, and then you should be able to answer them should you see this question on an exam. And we're running away from me here. Here we go. So the 2 questions that accompany this data, you can see the years there, so it goes through 2005, so it's now 2009 at the time of recording, so it's kind of old data. couple of years from now it'll be even older. But anyway, we still can interpret it within the context of the time it was collected. What can you say about cigarette smoking among high school students in the period from 1991 to 2005? And then the second question, which high school students are most likely to be smokers, and those that would be least likely to be smokers, from the panel, the graph on the right. Anyway, I appreciate your time and attention for me to complete the description of this very important portion of the nervous system.